Biosensors and BioMEMS, a survey of the present field. Introduction. Biomedical sensors and implantable devices employing microelectromechanical technology will be important to the future of biomedicine. Once just science fiction, the notion of a bionic man is now becoming a reality through the application of microscale technologies. Today it is not unusual to have a friend or family member who has an implantable device, such as a pacemaker, defibrillator, cochlear implant, biosensor, neurostimulator, or insulin pump. We have only seen the beginning of this technology development, and today's state-of-the-art implantable devices will be seen as crude and cumbersome tomorrow. With the rapid pace of development, it is probable that within the next 2 decades we will have as many medical treatments based on microdevices as there are pharmaceutical solutions today. This paper examines some of the most recent advances in the field, as well as the technologies that are likely to appear in the next few years. What is Biomims? Biomims stands for Biomedical Microelectromechanical Systems. It is a name applied to biological and medical devices that are created using advanced fabrication processes that allow the devices to be very small relative to comparable devices produced by traditional techniques. Biomims devices can also exploit the microscale to provide new functions that are not practical or possible in large-scale devices. The name applies to an exceptionally wide variety of engineered devices that derive from electrical, mechanical, chemical, and molecular engineering. The name distinguishes these from nanomems, which are submicron in scale, such as carbon nanotube structures. Recently, Biomims has become something of a misnomer, as many of the latest technologies are being designed and developed based on nanoscale technologies, which are many times smaller than microscale technologies. While current devices are manufactured mostly on the microscale, many of the functioning parts and the materials they operate on are at the nanoscale level. Nanomems for biomedical applications are mostly carbon-based materials that have emerged as prime materials because of their favorable mechanical and electrical properties. Carbon-based nanostructures such as graphene exhibit a high Young's modulus, stiffness, high strength, low density, low friction and large surface area. The low friction of a carbon nanotube allows production of practically frictionless bearings and has thus been a huge motivation towards applications such as nanomotors. Carbon nanostructures are much stronger than steel, which allows carbon-based materials to meet high-stress demands in biomedical applications such as weight-bearing prosthetics, like hip joint or bone replacements, where other materials would fail. The field of biomims encompasses micro-devices that are often but not exclusively made by the same photolithographic techniques used to make computer chips. Their applications include neuroprosthetics, sensors and actuators, and microchemistry systems. A microchemistry system, often called a lab on a chip, can analyze chemical properties of a very small quantity of material, such as a tiny blood sample. Advanced systems can perform several tests on the sample at one time. There are also drug delivery systems, miniature hearing aids, artificial retinas, DNA analysis systems, cancer diagnostics, and an amazing variety of devices which support the function of the human body. Figure 1 shows some devices that were developed by the Faculty of Biomedical Engineering at Arizona State University. Characteristics of Biomims. Conceiving and designing Biomims devices requires a different perspective of the physical world. These devices can operate on principles such as capillary force, van der Waals forces, and electric field forces. These forces become relatively strong when the size scale reaches very small dimensions. In the realm of the very small, the force of gravity is far less important than electrical charge and viscosity. From the perspective of microscale devices, engineers need to think about accomplishing tasks on an extremely subtle scale but with an exceptionally effective result. Biomims employs engineering sciences in ways that are more than just scaling down familiar devices used in biomedical applications. Rather, employment of new structures and new materials, including polymers and biological components, is necessary. In addition, we need to be concerned about things like biocompatibility, the effect of the device on body tissue, as well as the degradation effects of tissues and body fluids on the device itself. Development of a new product that is targeted for implantation into the human body is particularly expensive due to federal regulation of medical devices. Devices must be shown to be effective for their intended use, and above all, devices must be proved safe. Bringing a new device to market is not unlike the development of a new drug. In order to gain federal approval, there may be biocompatibility issues to resolve, clinical trials to perform, and Food and Drug Administration, FDA, requirements to satisfy. Categories of Biomims. We divide the Biomims field into several subcategories. Some devices are targeted to be implanted into the human body or are applied to the body. Many of these are being developed as alternatives to drugs which therapeutically treat illness or disease. There is also activity in using microscale stimulation devices to treat neurological disorders such as epilepsy and Parkinson's disease, and to treat neurological injuries such as stroke or trauma to the spinal cord. Another promising field of research is directed towards integration of the human brain with microelectrical components. For example, implants are being developed which allow volitional, thought, control of machines. Most of this effort is targeted towards rehabilitation of individuals who are quadriplegics in order to allow them some control over their environment. Then there are devices being developed for clinical applications such as rapid blood analysis, and there are those targeted for benchtop biological research. In this review, the focus is on specific microscale and MEMS-based devices that are used in medicine and biology. These include micromachines that interface to brain electrodes, blood glucose sensors, microfluidics, neural interfaces, neurostimulators, microbeam sensors. Several of these technologies have been the subject of research by the present author and by colleagues at Arizona State University. Biomims micromachines. Micromachines made by photolithography are among the most complex and sophisticated of all MEMS devices. They have seen some application in various forms of biomedical devices where motion must be achieved with an implantable device. Motion within the human body by a device is difficult since it implies sliding surfaces, a need for electrical power, and long-term reliability and stability. Biomedical implants are usually introduced by surgery and so once implanted cannot be easily removed for servicing. Only a few Biomims applications are well known for implanted devices, and these are used for positioning and repositioning of sensors. A specific example is application to brain electrode systems. Brain implanted Biomims micromachine neuroelectrodes. There are applications in biomedicine and research where very small electrodes are implanted into the brain. These microelectrodes are used in the cortex, surface layer, to detect electrical activity associated with the volitional desire to move some part of the body. Electrical activity is recorded as a very small change in voltage within the tissue. Near the top of the human head and about 2 centimeters beneath the scalp lie the parts of the brain where nerve cells, neurons, are found which control the muscles of the body. Specific locations in the cortex are associated with specific parts of the body. When a person moves a limb, there can be detected a corresponding electrical activity of these neurons. Neuroscientists have recorded these signals and developed a kind of map of the brain that defines what brain cells actuate certain muscles of the body. An interesting phenomenon occurs in which some of these brain cells become active even when there is intent to move a limb but no actual movement occurs. In a healthy person, the intent to move can be detected by measurement of microvolt signals from the brain cells about 120 milliseconds before any muscle movement occurs. In a person who has lost a limb or has become paralyzed, the intent to move can still be detected in the brain even though there is no limb movement. Thus, in principle, there is an ability to electrically record from the brain and determine a person's intent to move. Monitoring of the brain is accomplished by using an array of implanted microelectrodes whose signals anticipate movement of specific limbs. These signals can in turn be used to control machines. This is the idea behind advanced devices that allow quadriplegics to interact with their environment. Signals from implanted electrodes are used by computers to control robotic actuators. A problem recording the signals occurs because the body naturally tends to encapsulate the electrodes with scar tissue, meaning the electrodes lose electrical contact with the neurons. A solution to this problem incorporates a very small electromechanical actuator attached to the electrode. This device allows the implanted electrodes to change their position occasionally in order to continue to monitor neural events. By moving the thin rod-like electrode up or down a small distance after several months of implantation, the useful lifetime of the electrode array can be greatly extended. The Biomims device described here is meant to be implanted under the skull and on top of the brain cortex. It allows fine adjustment of less than a millimeter in order to make sure the electrode system, once implanted, is able to contact the desired brain cells even after the electrode has been encapsulated by scar tissue over time. Figure 2 shows a photomicrograph of a gear-driven micromachine that was made through collaboration between ASU and Sandia National Laboratories. The device is driven by an electrostatic vibrating comb motor. Under a microscope it can be seen that the combs move in an oscillatory fashion at about 40 Hz when energized. Electrostatic forces between two blade-like combs a few tens of microns apart are operative with about 15 volts as the electric field source. The neural probe chip enables precise bidirectional positioning of the microelectrodes in the brain with a step resolution in the order of 8.8 pm. The thermal microactuators allow for a movement of the microelectrodes of up to 5 mm in either direction, making it suitable for positioning microelectrodes in deep structures of a rodent brain. The rest of the mechanism converts an oscillatory motion to a linear motion through a ratchet-type configuration. Figure 3 illustrates the basic unit where there are two comb-like arrays on either side. These generate the actual force. They are connected to a cross piece that converts oscillatory motion in concert with a spring to a pulsatile linear motion of the vertical shaft. This connects to some gears which do a mechanical transformation. Fluidic Biomimes. The application of microscale fabrication techniques has allowed the manipulation of very small volumes of fluids on the nanoliter and even picoliter scales. This allows chemical processes such as separations, reactions, and analysis to be conducted with very small amounts of sample. According to a forecast by the Nexus Task Force, the market for Biomims is expected to reach $18 billion in 2005. The commercial success of these devices and the technical potential of other Biomims has driven research in a number of areas. As a result, over the past few years, Fluidic Biomims devices have become the largest and most diverse applications of MEMS devices. Fluidic Biomims now include drug-delivering neuroprobes, biosensors, general, bioreactors, cell handling devices, drug delivery devices, microchromatography systems, microfluidics, molecular detection/handling, neural interface devices, optical/retinal sensing, surgical devices, tissue handling devices,